Our keynote speaker today is Dr. Angela Belcher. Professor Angela Belcher is the James Mason Crafts Professor of Biological Engineering and Materials Science and Engineering at MIT. She is also faculty in the Koch Institute of Integrated Cancer Research. Professor Belcher is widely recognized as a pioneering leader in genetically encoded non-biological inorganic materials and devices. Her research focus has been to harness evolution for nanoscale control of assembly and growth of technologically important materials, enabling an environmentally sustainable approach to manufacturing. Her methods permit increased scope of elements and compounds that these biological molecules can interact with well beyond their natural history. She has demonstrated useful application of this technology to batteries, solar cells, fuel cells, environmental remediation, heterogeneous catalysis, photocatalysis, carbon capture and storage, and medical imaging. Professor Belcher has received over 50 awards for her work. Her publication H-index is 54, including 4 papers with more than 1,000 citations, and holds 30 issued patents along with 25 more pending. She founded 2 materials companies that are producing products. Cambrios Technologies, now Cambrios Materials, has had 30 products in transparent conducting display manufactured by NEC, Huawei, and LG. Siluria Technologies has 2 pilot plants producing ethylene and opened a demonstration plant in 2015. Siluria is currently preparing for worldwide production. Professor Belcher has graduated 42 PhD students who have gone on to work in academia, industry, technology transfer, government, and policy. She has been very active in undergraduate teaching for 17 years, teaching in chemistry, materials science, and biological engineering, maintaining a 6.4 out of 7 student rating over the last decade. She has mentored 162 total undergraduate engineering semester projects at MIT. She has been extremely active in K-12 educational outreach at the national and local levels. Please join me in welcoming Dr. Angela Belcher, who will speak to us about engineering biology to address some of the world's biggest challenges, including energy, the environment, and medicine.
Thank you. Well, thank you very much for that very kind invitation. I think that was my whole lecture, so I don't have to speak now. But it's a huge honor to be here, and congratulations to all the participants and their parents. I'm just so proud of you guys, and I think it makes me feel really good about thinking about our future. What I'm going to talk about today is some of the research that's been my passion pretty much since I was in middle school. When I was in middle school, I got really interested in where did the first replicating molecules come from? How do we go from really small molecules to large molecules to whole cells to human beings? And when I went to undergraduate, there was no major in that, and so I put together my own major between chemistry, biology, physics, and geology. And I've really followed that love the whole time. And so part of what I wanted to share with you today, I'm not a mathematician. When I was asked to speak today, I was very honored, and I said, oh, well, I'm not a mathematician, I'm an engineer, but maybe that's applied math. As we heard, MIT's name might be changed, and I'm okay with that. But I thought I'd tell you some about what I'm most passionate about and what we're doing in making materials to try to make the world a better place. And I like demos a lot, and my students usually set it up, and as you go to graduate school, you'll realize that all the really hard stuff behind the scenes, that's all students. It's not faculty. And the fact that I set this up by myself today, my students are going to be very, very proud of me when I send them a picture later. But I run a big group with students from all over MIT campus. I don't really say I want only students from material science or chemistry or biology or biological engineering or chemical engineering. What I look for are students that are really good in one thing and are passionate and are interested in learning basically how to integrate that into many other topics. So I love teamwork. And this is our Halloween party here. I'm not sure, we're dressed as a movie. I'm not sure what movie it is because I didn't see that movie, but we're supposed to be like cool spies, I think, for our annual Halloween costume. The other picture I have up there is Professor Paula Hammond, who's chairman of chemical engineering, and Professor Sangeeta Bhatia, who's in electrical and computer science and engineering. We're holding signs that says, I look like an engineer, because we're part of a campaign to reimagine what an engineer looks like, which was really fun to be a part of. So I've spent my career studying nanomaterials. And everyone here knows nano, and they don't even make nano iPods or what are they called? iPhones anymore. But I'm interested in materials not because they're small. I'm interested in materials because when you take bulk materials, I mostly work on semiconductor materials, inorganic materials, and you either grow them from a couple of atoms at a time and grow them bigger and bigger and bigger, but still staying at the nanoscale, the properties change. The properties of materials at the nanoscale, when you have collections of hundreds or just 1,000 atoms, are different from bulk materials. And we all are nanostructured materials. If you think about the size of our cells and the molecules inside of our cells, all those molecules are molecular and nano-sized materials. But I'm interested in controlling them using biological methods. And so I brought a demo here. It doesn't look very, very fancy. Actually, this is supposed to be graphene. Anyone ever heard of graphene? It's kind of a super material right now. Graphene is made out of carbon. I'm not sure, I was never brave enough to take a math prize exam like you guys, but did you do it on pencil and paper, or did you use, was it done on computer, or how was it done? Pencil, okay, all right, so when you take your pencil and you're doing your calculation and you're sliding it across the paper, what you're actually doing is pulling graphite across the surface of your paper. And if you took that graphite and you pulled off one small layer of it, one atomic layer, that's what graphene is. Graphene is basically atomic layers of carbon. And these atomic layers of carbon have really, really interesting properties. And so there's been a Nobel Prize in the graphene-based materials. It has interesting optical material properties, it has interesting electrical properties. But one of the things I'm interested about, when you take graphene and you roll it like this, it becomes a tube. It's called a carbon nanotube. Anyone ever heard of a carbon nanotube before? Another kind of super material. Basically, they're about a nanometer in this direction, and they can be hundreds of nanometers in this direction. And they're also kind of a super material because they have interesting optical and electronic properties. But they're very, very small. Imagine going with some forceps and trying to pick up a 1 nanometer object. Very, very difficult. And so what we've done in my lab is we've tried to genetically engineer viruses to pick up really small things and move them to where you want them to go. Now, many people in the audience may think, oh, viruses, those are terrible. I get the flu vaccine every year to prevent viruses. But we look at viruses as also kind of beautiful. We look at them beautiful for their molecular structure. And the kind of virus that we use is called M13 bacteriophage. Anyone ever heard of bacteriophages before? Yeah, so bacteriophage means bacteria eater. They infect only bacteria, so they're not toxic to humans. And we use them to do things like build batteries and build solar cells and build catalysts. So when I was a young professor a long time ago, when I first started, I wrote my first grant proposal, which is I asked the government for money to do research. And I said, I'm going to genetically engineer viruses to build better semiconductors for computer chips. I wrote it off, I spent a lot of time, I put my heart and soul in it, I sent it off to be evaluated, and it came back with one line, "She is insane!" exclamation point. Now I took this very, very bad, and I was kind of crushed at first, but I really thought that viruses can make electronic materials. And why did I think that viruses can make electronic materials? I thought viruses can make electronic materials because of these abalone shells. I heard some people from California, I'm not sure which part of California, but abalone are really common off the coast of California. Well, abalone are shells that are made out of calcium carbonate. They're basically a biocomposite material made out of 98% by mass calcium carbonate, but 2% by mass protein. Yet they're 3,000 times tougher than their geologic counterpart. Okay, that's really crazy. The geologic counterpart, like chalk. You guys probably didn't use chalk on your exam, but basically, they're 3,000 times tougher. So what's the difference between chalk and this? I'm going to throw this to you guys. You guys can pass it around. Don't be nervous. Good catch. These have about 500 million years to get good at making these structures. So they have about 500 million years of evolution to learn how to make calcium carbonate-based materials. but also that they're made out of nanomaterials. So chalk is not a nanomaterial. This is a nanomaterial. I said nanomaterials have interesting optical, electronics materials. We also have interesting mechanical properties. One of you guys are good catching? Oh, good catch. One more over here. This is a small one. I'm not sure how it's going to fly. OK, no injuries. No one was injured during this presentation. OK, so here's the thing. Organisms in the ocean already make hard materials. They make bones, calcium carbonate. They make silica. But they haven't used the rest of the periodic table. That's why you got the periodic table. So what I said as a young professor is, what if we give biology the opportunity to use the whole periodic table? What can it do, and how do you do it? So that's what I'm going to tell you a little bit about today. OK, so I'm an engineer. I'm focused on problems. I'm only interested in really solving problems. And the problems I'm interested in are food, water, energy, and healthcare. And everything we do in our lab is trying to focus on trying to solve the problems that we think are most pressing to the planet. And I think that many of these problems have scientific and technological solutions. Now, everything we do is based on how nature makes materials. That's that abalone shell that's hopefully being passed around. If you want to keep it instead, I guess that's fine. I'll show you some more after here. Or things like plants, which use photosynthesis to make their own energy. I quickly mentioned a nano, and nano is cool, and nano is small. But the most interesting thing about nano is not really that it's small, it's that its properties change. The way, if you think of this as a nanoparticle with only thousands of atoms, the way those atoms interact and the way that they feel each other changes at the nanoscale. And you can tune those very easily. And by tuning them, you can change their properties to make new kinds of devices and things you might be interested in. So just to give you the idea of DNA is about 2.5 nanometers. Compared to the size of a bacteria, about 2.5 microns, compared to a large raindrop, 2.5 millimeters. The carbon nanotubes I mentioned before, about a nanometer in diameter. And then gold nanoparticles, about 4 nanometers in size, about 1 million times smaller than this ant. And so nano is great because it's small. Nano is not great because it's small, because it's hard to move small things away. So my research focuses at the interface between nanotechnology and biomaterials, because that's how biology learned how to make materials, like our bones are nanostructured materials. Those shells are nanostructured materials. Diatoms, which make glaceous structures, are nanostructured materials. And so we work on trying to change nanomaterials and biomaterials for energy, healthcare, and the environment. And we do it by controlling the size of the particles, controlling their placement. And so then we work on all these different areas, from solar cells to batteries to new kinds of fuels, water purification, therapeutics, and imaging. And again, people still say to me today, they say, Angie, why don't you focus on less things? You focus on way too many things. And I said, I focus on just a few things. I focus on using biology to make better nanomaterials so that those nanomaterials can go out and make new devices to try to make the world a better place. So the Soluria Technologies is one company that I started. Cambrios Technologies is another. And 2 other companies I'm working on standing up right now are Geyser Biotech and Nirvana Diagnostics. One is for energy and the environment, and the other one's for cancer diagnostic and imaging. And so when I showed you that abalone shell, and I told you that abalone shell was wonderful because it's 3,000 times tougher than its geologic counterpart, it's made in ocean temperatures and ocean pressures. So it doesn't use toxic materials. How do you think those cell phones that you probably hopefully have turned off right now, how do you think those are made? Are those made in low temperature, low pressure, environmentally friendly conditions? They're not. But biology's evolved to do that. When they make shells, when we make bones, when the picture on the bottom with the star-shaped structure and the things that look like spaceships, that's glass. That's the way biology makes glass at ocean temperatures and pressures. It doesn't put toxic materials back into the environment and doesn't use toxic materials. And so we said, wow, what a wonderful idea. Why don't we manufacture everything using environmentally friendly synthesis of materials? And so when we think about future technologies, we start about 4.5 billion years ago, the beginning of Earth. It only took about 1 billion years to have life on Earth. But all of a sudden, you had really fantastic things happen, like photosynthesis. I'm sure all of you guys could diagram photosynthesis for me if I asked you to. But it's an incredibly complex reaction to take sunlight and to interact with proteins and other kinds of molecules and to make your own energy and food. So but it wasn't until about 500 million years ago, during where that red line is, that's the Cambrian geologic time period, when organisms started making hard materials. All those billions of years, everything in the ocean before were soft and squishy. Okay, it's kind of crazy. What do you think it's easier to do? Make photosynthesis work or build calcium carbonate, build chalk? Well, actually, photosynthesis happened much, much earlier. And the reason is because it was during this time. Have anyone ever heard of the Cambrian geologic time period? Yeah. Cambrian geologic time period, you think about the explosion of life, all these interesting things happened. New organisms started appearing, making new kinds of structures. Well, what happened during that time, there's increased calcium and iron and silicon in the ocean. Remember, all these organisms are living in the ocean, and it became toxic to them. What they did was they learned to take the enzymes and structures they have and push it outwards and start building shells. That's how shells started getting formed, and that's how diatoms started getting formed. And what I said was, well, I thought life did a pretty good job, but I think life could have done better if it would have had more opportunity. And so what we wanted to do was to train simple organisms like bacteria and viruses to build things like batteries. Again, I've been given a really hard time. You can't build a battery out of a virus. Well, this is a battery that's a virus-based battery. It's an all-virus battery. It's built. It's non-toxic to humans. It went to the White House for a press release with President Obama. I wasn't invited, but the battery was invited. I watched it on closed-circuit TV. So my students and I were really, really proud. But people said, there's no way that you can do this. And basically, if you think about what you've learned in your biology classes, about the molecular biology you've learned, and what's the amazing things that are happening in DNA research and genetic modification, you can basically train your viruses to build batteries, and that's what we did. And we built our batteries at room temperature. We didn't use toxic materials. It's all made out of natural materials. Okay, so basically, if you think about, you say, why biology? If you think about the size of the kinds of molecules you've already learned about, DNA and proteins and ribosomes, they're all at the nanometer scale already. So you can use those as your template, basically, to start building materials. So we look at the whole of biology, whether it's proteins or polysaccharides and sugars or nucleic acids or antibodies or protein cages, as a molecular toolkit to build anything we want to be able to build. Here's some natural materials. This is a coccolithophoriid. It's a unicellular algae made out of calcium carbonate. Next to it's that abalone shell. And if you take that abalone shell and you break it and you look at it in scanning electron microscopy, how many people have done scanning electron microscopy? I'm going to be amazed if anyone does. So high-resolution electron microscopy. You can see what its structure looks like. And it basically looks like this. It looks like these brick walls that are actually intercalated into each other that gives it this really special structure that makes it very tough and strong. Diatoms make glass, and there's organisms that are bacteria that make magnets. Anyone ever heard of magnetotactic bacteria? Bacteria that make small single-domain magnets used in navigation. They make them better than we can make them for storage technology, for data storage technology. But the key to them not only is the fact that they're nanostructured, but between every one of these little individual calcium carbonate structures, they have proteins. And these proteins, which looks like the Swiss cheese right here, actually controls their structure exactly, which makes them 3,000 times tougher. Now, when I was a young professor, too, I worked in the black pearl industry. And these are black pearls because calcium carbonate that make shells are similar to the calcium carbonate that make pearls. I don't know if anyone has any pearls on. I don't have them on. But they're nanostructured materials. The reason they're so beautiful is because proteins control them to make these perfect nanostructured materials. So my job, it was a great job, which was to see what's the difference between a $3,000 black pearl, or more expensive than that sometimes, and like a $30 black pearl. So I would take a diamond saw, cut it open like $3,000 black pearl, and then try to understand what was so great about that nanostructure compared to the one that wasn't. And I should say that it was a pretty good first job. And the black pearl industry paid a lot better than being a professor, and it was kind of fun. But basically, the way that an abalone grows a shell is it has this protein. And if you take your periodic table and you flip it over, you'll see the amino acids with their pKa's. I'm sure everyone has studied their amino acids already and their pKa's. And this is if you're on the subway or something and someone asks you, what's the pKa of aspartic acid or something, you could just, if you don't have it memorized, you can whip out your periodic table card, pull it out, and impress your friends and neighbors. But basically, these proteins that are responsible for making the shells are very anionic. They're negatively charged. They're rich in aspartic acid and glutamic acid, so they're negatively charged. They can grab calcium out of the ocean and start building calcium carbonate, an atomic layer at a time. And that's the way they do it, and there's a little cartoon here. That basically the structure grabs onto the atoms, starts to build calcium carbonate, and put them down. And to build structures like this. And so what my lab has spent the last couple of years doing was trying to say, okay, biology did a pretty good job. It used calcium, it used silicon in the form of silica, it used iron, it used a couple of other elements, but didn't use the whole periodic table. What if organisms could genetically encode the whole periodic table? So I was really lucky that a couple of years ago that President Obama came to visit my lab. And he came to visit our battery lab and our solar lab, as well as other labs at MIT. And I have this MIT periodic table made for the freshman class, and it says, "Welcome to MIT, now you're in your element." And I did that because when I came to MIT, I felt like I really fit in. I felt really at home here. And so I wanted everyone else to feel the same way. And so it says, it's periodic table on one side and the amino acids for the PK on the other side. So after President Obama had visited, I wanted to give him a periodic table. And I wasn't sure how to bring that up in conversation with the president. And so I started, "Sir, I'd like to give you this periodic table in case you're ever in a bind and need to calculate molecular weight." And he's actually a really clever and funny guy. He said, "Thank you, I'll look at it periodically." And later, he gave a lecture, and he said, those MIT people give a periodic tables, what's with that? So I definitely have one for all the students and maybe some of the family too. So if you didn't get one, come get your presidential periodic table after the end of the award ceremony. And so that's what I spent my life doing, is trying to give genetic information in non-living structures. And the basic idea is that it's happened naturally through evolution. It takes about 50 million years to get good at it. So at MIT, almost all the undergraduates work in labs. From maybe their freshman year to their sophomore year. Many students will do a senior thesis. And I've never got a student to do a 50 million year project, never once. I mean, our students all graduate in time. So you have to come up with a much more rapid way of developing a protein that can build a material of interest. And so what we decided to do was borrow an idea from the drug discovery industry called phage display. Where we go in and we take this, my students made this for me. This is a model of the M13 bacteriophage. You can see here that it actually has proteins, has 3,000 proteins along the outside of it. Anyone want to guess what the structure of these proteins are? These are alpha helixes. So it has 3,000 beautiful alpha helixes around the outside. It has single-stranded DNA on the inside of it, and this is the DNA coding for 3 genes. And I'm going to see if I can work it by myself. This is going to be iffy. But the genes, here's one gene here that codes for this protein. Couldn't exactly do it. This protein here codes for these proteins. And this gene here codes for these proteins. And so what we can actually do, it works. OK, I did it. Yeah, OK, isn't that impressive? OK, so what we can actually do is take that single-stranded DNA and use molecular scissors to cut it. What's another word for molecular scissors? How are you going to cut DNA? You're going to use enzymes, right? You're going to use enzymes that go in at particular places, break it apart, and put a random DNA insert in it. And from that random DNA insert, you could actually start putting new protein sequences of interest in there. And so we think of a virus as a molecular toolkit that can easily go in and cut and paste and add what you want to add in and make genetic changes. And so using this process called making libraries. And so what we have is we have a library of viruses that are all genetically identical to each other, but they differ from each other based on a small sequence. So we have 1 billion viruses in way less than a milliliter solution. I don't know which kind of virus is going to make the best battery. So I'm going to take 1 billion viruses that are all slightly different, force them to interact with a battery electrode, and see which one works the best. From that billion, I may have 1,000 that can interact with that one. I say, pretty good job. It's kind of a Darwinian process. I say, of that 1,000, which one can do better? And then I'm looking for the 1 out of a billion that can build a better battery, the 1 out of a billion that can build a better solar cell, or 1 out of a billion that can detect ovarian cancer much earlier. And that's basically, I think I have like 98 more slides, but that's basically what we've spent the last 15 years doing, trying to give genetic information to solar cells and batteries. So I showed you that battery that we have. Right now, we're building batteries to put in unmanned vehicles for trying to double the time of a drone flying. We're building viruses that can convert natural gas into ethylene, so C1 to C2 chemistry, from C2 chemistry into drop-in fuel replacements at 60% reduction in carbon footprint. And we did that through selection and evolution. So right now, in one of the companies I founded, we're making a ton of ethylene a day at 60% reduction in carbon footprint. And we figured that out through selection and evolution. We're genetically engineering yeast, the same kind of yeast you use to make bread or beer or wine. And instead of making bread or making wine, we're having them clean up radioactive spills. And we're having them clean up toxic metal sites as well. And then the newest area that my group is in is we've been genetically engineering these viruses to pick up carbon nanotubes like this. But actually, one thing I didn't tell you about carbon nanotubes is that carbon nanotubes give off light at a really particular wavelength. And when you engineer them correctly, they light up. And they light up in an area that's invisible for us to see, although you can see this, but that we can see deeply into the body. And so now we've engineered viruses to pick up carbon nanotubes like this, find ovarian cancer tumors like this, and sit on them, and give off this light in a special wavelength. And then we've developed with Harvard Medical School a new imaging device to help surgeons we're still in animal models, help surgeons find tiny tumors for ovarian cancer. So ovarian cancer tumor size is usually diagnosed at 1 centimeter, but we can see 0.2 millimeters in size using this new technology that we developed. And we developed the whole imaging system to try to help surgeons, when they go in to do ovarian cancer, find and remove tiny tumors. The initial data we have in animals, not in humans yet. We hope to be in the first human trials in about 1.5 years, is we're seeing increase in 40% life in our animals using this guided surgery. So I guess my message is to you is that I hope you can tell I have a really good time with my job. Every day I come to work and I work with really smart people, work with really smart students and other faculty, and every day we come together to think about how can we solve problems that we think are important to the planet. And did I have this master plan when I was always interested in the origin of life. Did I have this master plan when I started? No, I actually went to undergraduate for 5 years. They kicked me out. They said, you have to graduate. And I said, this is the best job in the world. I could do this forever. And they go, no, this is not a job. You have to go on and do something else. So I said, okay, I'll go to graduate school. And I went to graduate school and I finished and I go, oh, this is the best job in the world. And they go, being a professor is very similar. So I became a professor, and I've just studied what I love my whole life. And I hope that you're finding that passion, and that you continue to be passionate and find what you love to do. And I hope that you use that to try to make the world a better place. So with that, I would like to, it's a huge honor to be here. You guys are all, fantastic. And I hope to see you more in the future. Thank you.